Welcome to the Supplement Engineer Podcast. My name is Robert Chenesky. Joining us, industry veteran, expert in supplement science, sports nutrition, and a bunch of ingredients that I'm excited to talk about, Dr. Hector Lopez. Long time coming, Doc. Thank you so much for joining us. We've been I don't even say colleagues, associates. We've talked for a couple of years now, and I'm so excited that we finally get to do this interview. So thank you for joining us, first and foremost.
Yeah, no, it's a pleasure finally being on. You're right. It's been a long time coming. And I think it's safe to say we're colleagues and friends, industry friends. So, yeah.
Yeah. Well, yeah, I'm always cautious because I'm on the podcast with an MD guy that's like on the cutting edge of supplement science and research. And so I'm the little geek that's at home, looking at all this from the bystander thing. So it's like, I'm very cautious with using those kind of terms, but it's a genuine pleasure and honor to be able to speak. So thank you.
Thank you, man. That's very kind. Thanks for the kind words.
Just as a brief recap for the audience that may not be as familiar with the stuff or may not be as nerdy with the ingredients as I am, just as a highlight, we're going to say 3DPump, VasoDrive-AP, Alluvia PurpleForce Tea, SmartPrime-OM, which is the new omega-3 that you just got released. I don't know which other ones you can divulge at this time. TeaCrine, Dynamine.
Yeah.
Anything else you want to leave?
Yeah, there's a longevity one, NAD3, that's been kind of in, I guess, semi-stealth mode, kind of like SmartPrime has been over the last couple of years.
Mm-hmm.
But it's probably about ready to see the light. A few more, old school going way back. Obviously, HydroMax was probably the first one that really made its way into the industry as a branded ingredient for us.
Right.
It was called something else before HydroMax when we were just, basically stabilizing glycerol and felt like, that was like 12, 13 years ago. So
mm-hmm.
long time ago. Yeah.
Yeah, back to the old glycerol monostearate days where you get those giant tubs of clumped up powder that just ruined your entire tub of pre-workout.
Yeah, the wax, right? And then even once you got it into your shaker, it would just like sit on the surface. It's a nice waxy coat. Yeah.
Yeah, exactly. So you started off in sports medicine, you pivoted into supplement science. Have you gone back to, when you were a kid and a teenager, were you always active in athletics? Did you have a passion for science? So how did we get from the young Dr. Lopez to the grizzled veteran that you are today?
Yeah. I've definitely always had a passion for both nutrition or anything that I could read on a label, which sounds kind of weird for a kid. But I think, to be honest, it all started because I was allergic to egg, ovalbumin, basically. So egg yolk, egg white. Weird. From the time I was 1. So the first time they introduced it, parents probably should have introduced it like they do now much earlier. So I would have been desensitized and tolerated it. So I was 1 and I was hospitalized. From that point forward, I had a pretty substantial egg allergy. Mm-hmm. And in the 70s and 80s, there was a lot that egg was used for as an emulsifier. It still is, obviously. But from the time I could start reading, I would just pick up anything I was consuming or in the grocery aisles and just read the ingredients, because my parents kind of taught me from an early age, look, this is what you look for. If you're going to eat something at school or someone's going to give you something, if you're away at a friend's house, this is what you look for. So I guess that was probably it's kind of, probably a little weird, but it's probably the genesis of, if I think like what has had me come full circle, that's probably the genesis of what got me interested in ingredients, bioactives, functional ingredients, formulations, if you will, because I was reading whole ingredient labels, from the time I was probably 6 or 7 or 8, So that started me along the path of just awareness of, what are these ingredients? Even some ingredients you couldn't pronounce that most people, would say, well, those are additives or excipients or whatever. I was already kind of reading them and at least aware about them.
Yeah.
And then what happened is then there was the intersection of that with the fact that my dad he was an avid collector of all the old, fitness and bodybuilding magazines and, the old school Muscle and Fitness, old school Flex. He was a commercial storyboard illustrator, and one of the So what he would do is they would hire him to write out storyboard frames for like a commercial that would air on TV for a big brand or something.
Yeah.
And so the art director would hire him. He'd illustrate out the frame by frame. And so I remember Arnold and Sly Stallone and Carl Weathers, the old Rocky, and obviously that whole sort of team, Dolph Lundgren eventually. So he was doing storyboard frames with a lot of these characters, and then it just honestly, I would read some of the Muscle and Fitness like cover to cover, eventually like Flex, and then Muscular Development, and then it got all the way into the 90s when I was in grammar school and then high school.
Yeah.
And then growing up, I loved playing baseball. Baseball is, I'm Cuban. I'm first generation Cuban American. My parents are both Cuban. So baseball was my diehard passion. I wanted to be a pro baseball player. Who didn't, right?
Right.
And then I was, I guess, aware enough to realize I probably didn't have the attributes and the talent. And I could work my ass off, but the chips were going to be stacked against me. So then I quickly transitioned into maybe wanting to explore how else I could get involved in pro sports or pro baseball. Then I remember my dad mentioned to me, he's like, if you did like something like communications and maybe a sportscaster, in journalism. And then I kind of explored that a little bit, but then I always kept coming back to the like human performance of athletic feats always fascinated me. But I always wanted to know why and how. And bio and chemistry, and eventually as I got older, biochemistry and really digging deep into mechanisms. That's ultimately what really just, it's the kind of thing, sometimes they ask like, if you really want to know like the 3 things to look for when you're looking for a career. And I credit my dad with really, giving me this sort of wisdom. He passed away last year. So, but he always told me, he said, 1 is, what do you love to do? 2 is, what sort of comes naturally to you? What sort of proclivity do you feel like you have? And then 3, what do you feel like the world needs from you, or what you can contribute to the world? And if you can find the intersection of those 3 things, and there are many ways you've seen this, like Ikigai, right? The Japanese sort of philosophy. Right. If you can find the intersection of all these things, then you're probably going to be living a fulfilled life. And that was really all like my parents did. Like there was never the pressure of like, in some cultures, it's, you should be a professional, either an engineer, a lawyer, a physician, right? That was really the case. It was always do what you love, do what you wouldn't mind doing on a Saturday night when your friends are asking you to go out, and you're like, you know what, I could do this instead, and you wouldn't mind doing it. You don't feel like you're missing out.
Right.
If you find something that, kind of fans your flames that way and fuels your fire, then go after that. And that's ultimately how I found my way into the end of high school, going into undergrad. I ended up wanting to get really, really fascinated by molecular biology and recombinant DNA technology and molecular genetics. So I ended up, I went to Rutgers, and I double majored in biotech, molecular biochem, and then also nutritional sciences. So that was the combo that started it all, I think, early on. And then I minored in exercise physiology. So that was, the dream, like as an undergraduate, granted, right?
Yeah.
But as an undergraduate, I felt like I got a chance to put together like everything I loved in the sciences and in a vocation. So that's how it started. And then I went through undergrad, continued to find out how I could get involved. Like I started sort of going to the local ACSM chapter meetings. I started meeting people there. NSCA, I started meeting people there at the state level. Then I was asked to present when I was still an undergrad. So I still didn't have my CSCS, because you need a bachelor's right before you can qualify.
Right.
I still didn't even have my CS, and I remember presenting for one of the state, meetings in New Jersey. And then I at that point, late in my undergraduate years, my dad ended up having some health challenges that kind of, he got a small trochlear nerve palsy in the eye. So he developed double vision from a small stroke. And as an artist, like that's your livelihood, right? So, so that brought me really closer to medicine for the first time. And that was the first time I ever considered, I said, I think there's a potential here for me to bridge the gap, because I felt like there was so much that medicine was missing. This was in the 90s. Even though you could consider a lot of nutrition science still in its infancy back then, there was still a lot that was happening, advances in nutrition and natural bioactives and even just exercise science principles that were not being applied in the clinical realm in conventional medicine. And I thought there was a huge divide there, these two things on separate islands. So that's when I decided, I said, you know what, Dad, I think I'm actually going to pursue medicine. And so I approached my counselor, said, well, there's, MCATs, you could take the MCATs, you're already, a senior, you could take the MCATs in like a month. You probably want to prepare more than just a month. I took a practice and I did pretty well. So, and then I ended up, pursuing medicine at that point.
Fascinating. There's a lot of different avenues we could take. We could start with you with the ingredient stuff, but I almost kind of want to start with just, I guess, high level, just research questions, because this is the opportunity to be able to pick the brain of people that are actually designing the studies for the ingredients that we've come to know and love in the sports nutrition space. Because we see a new trademarked ingredient or a new branded ingredient at least once a month, if not more frequently than that. And quite often it's backed by one study that may be good, maybe not be good. And so for somebody that's actually kind of in the weeds, that's either exploring these ingredients and bringing them to market, or has been contracted as part of the research firm to design these studies. Let's start there with, how does that first start happen? I guess we can limit it to your case, because you probably had it both ways, where you stumbled upon the ingredient and then you want to start working on bringing it to market, or you've had a company say, hey, we know you're respected in this space, you know how to design a study and carry it out. How do we do this in an ethical way to where it comes across to the consumers as this brand, this individual, this research study, it's on the up and up? Because we've seen ingredients come to market in the past, and the researchers behind them have juked the stats, and we don't need to get into specifics or anything. Like you understand what the case that I'm speaking of, more or less. So I guess let's start there. How does that first start? Does somebody like Compound Solutions, Nutrition 21, Creative Compounds, any of these brands, do they pitch you, or do they bring you in on that initial stage when they're first vetting an ingredient?
So we could start there before segueing into, because we sort of like to keep a pretty strong Chinese wall, if you will, a firewall between ingredients that we have an IP interest in, right? Clearly, we would have a conflict of interest if all of the data on that particular ingredient was only coming out of our CRO or our lab. Correct. And we recognize that, obviously, but we do have an advantage in that we can use our own CRO as an incubator for ingredients or compounds that we've screened or we've managed to find via in silico methods or machine learning or AI, and we want to explore deeper. And so it allows us to do that. But that we operate that as a completely separate business from our CRO. And the way we started running the CRO, Tim and I partnered up almost 12 years ago now, I'd say 12, 13 years ago. Okay. So I think it for some people, we continue to stay oftentimes in the background. We don't do any outbound marketing. There are, the majority of CROs do have like a marketing department. They've got a sales team, right? They're actively promoting their services, and they're actively trying to secure leads for clinical studies from a lot of branded ingredient houses.
Right.
We did things a little differently. We just felt like we could start growing things more organically and just word of mouth, because then we can also control the growth as well, as opposed to like putting huge dollars in marketing and then being inundated with all these requests for proposals, and then maybe having the quality of what we wanted to put out there as far as evidence-based supplement science is concerned. So we'll start with like what got the CRO started, which was just, providing a good, solid service as a full service CRO, right? So there's some CROs that what they do is they sort of, they'll take your concept or your design and they'll try to sort of place your study at various sites within the US or Canada, mostly within the US. Sometimes they'll try to place your study in India or in other countries as well. And then,
is there a reason for that? Is it just cheaper to outsource it to other, or is it more friendly research teams kind of thing, or?
A little bit of both, but I think it's often comes down to the economics of it. Yeah, so there's a couple of different tiers when it comes to the economics of running studies. I would say a CRO like ours is probably middle tier as far as the cost of running studies within the CRO business. Okay. Because we're a very lean team, because we don't have we don't do outbound marketing, right? So we don't have the overhead that some other CROs might have, right? Because all we do is word of mouth, and we work long term with a lot of our partners. So once we do a study with a sponsor, we usually end up doing multiple studies. We develop a strategic partnership or relationship with that that research sponsor. But I would say, so you could place studies at various sites. And yes, the economics of being able to run studies overseas oftentimes outweighs the potential upside of being able to say you ran the study in the US. Now, what's nice about running studies is whether you do it overseas or you do it in the US, like the key is really you can find a lot of what you need to find by going first and foremost to the methods section. And that's oftentimes the last section, or even a section that most people don't ever read, right? They're seeing headlines and abstracts usually, right?
Correct. Yeah.
And so you can learn a lot by just going straight to methods section on every study. And that's often what you'll see a lot of the nuances popping out, like, okay, so this study was done overseas in a certain country where, well, why was this in the study with 34 men? Their average BMI was 22. They weighed about 72 kilos or 65 kilos. And they're novice as far as strength training background is concerned, or training experience, right? They're untrained effectively. And we're wondering why we're seeing these, and then percentile versus absolute changes. And we're wondering why we're seeing these changes, a 45% increase in one rep over an 8 to 12 week period. Yeah. And again, the devil's in the detail. So if you look at the methods section and you start scrutinizing it a little bit, you can start figuring out, okay, I see what happened here, right? It would almost feel like if we were here at our CRO and Tim and I would say, hey, let's go to the local grammar school or 8th grade high school or even freshman high school class. We recruit all our subjects from there. Because they would make great subjects for a study, right? If we're looking for performance improvements, they're nowhere near their asymptote, right? In terms of their genetic limits or their training background. So, I think that's critical. To your point, I know you're obviously much more attuned to being able to critique studies and pick them apart a little more than most, right? In the industry. So, but with us. The way it starts with us is usually an initial relationship with a sponsor, like you mentioned, like a Nutrition 21 or a Compound Solutions, and they've got an ingredient. They bring us the ingredient. We usually already know the ingredient. We usually already pre-firm on the science, like the mechanisms of the ingredient, but we'll do a deeper dive. And then we'll often work backwards. We'll ask them, in a perfect world, if you could have any endpoint, and what claims do you ultimately want to make in your marketing, on your label, your messaging? Then we talk about what your consumer, target consumer avatar looks like, or who you want to go after. And those are usually the things that allow us to now start working backwards in designing a study that would ultimately help them substantiate those claims, that in a perfect world they'd like to be able to make. Again, whether they're exercise performance claims, body comp claims, or some other health category, claims, whether it's immune, longevity, joint health, cardiovascular, endothelial health, name it, et cetera. So we usually work backwards. And then, of course, the major parameters that I think often don't get enough credit is just like, like when you're formulating, you've got huge constraints on cost of goods.
Yep.
Think about it from our standpoint as researchers. We also have cost constraints as well with every study we run.
Right.
And I think that oftentimes, kind of either goes by the wayside, or there are many armchair researchers who've never been in the trenches, never actually run an ICH GCP guidelines-based study. And for those who may not know, those are, these are global standards that are recognized, like International Conference of Harmonization, good clinical practices, just like there's GMPs for manufacturing. There's GCPs for running clinical trials, and there's something called consort guidelines. So we run most supplement studies effectively the way you'd run a pharma trial, within reason. Of course, there's still some things that we couldn't do that we would be doing in a pharma study. And we do run occasional pharma studies as well, where we're just a site for those studies. Those studies already come with a protocol, like a full, 65, 70 page protocol intact. Everything you need from an inclusion, exclusion criteria, the design, the primary, secondary, tertiary endpoints, how you're going to manage and track adverse events, safety. And then that, all the PI packet already, it all comes ready for you to just take it on as a site if you feel like you've got the patient population to be able to take those on. But we run all our supplement studies the way you'd run a pharma study within reason. And usually it's the scale, right? It's the sample size number that ends up limiting you, and the number of total visits to the lab, right? Because those are things that will often drive cost.
Okay. One of the ingredient houses come in, they have the design protocol. Do y'all get any input on to say, hey, maybe these metrics that you're telling us to run or assess aren't the most valid? Now, obviously, it's in the ingredient house's interest because they're going to set it up a certain way to where hopefully it works out, because they don't want to pay, $50,000, $100,000, $200,000 for the study and it come up, blank, or it shows that the ingredient's not as effective as placebo or something like that. Do y'all have any input in that instance to where you can kind of push back or just give constructive criticism and say, hey, this is a more accurate or a more genuine way to assess whether this ingredient is doing X, Y, and Z or not? And then are they receptive to that, or they kind of say, hey, just here's our money, run it like we tell you to. If not, we're just going to go somewhere else?
Yeah, no. Actually, we would probably, and that's one of the benefits of running a tight ship the way that we've been able to run it, is we would probably end up, how like sometimes a physician has to quote unquote fire a patient. At that point, we would probably say, look, we're probably not the CRO for you to work with. It's oftentimes that when it comes to research design, we're the ones having the primary input, given, their budgetary constraints and given, what they ultimately want to say about their product, because that's their product, it's their IP. We have to respect that. And then we can have discussions about pros and cons and trade-offs of using a particular instrument or an outcome measure. And sometimes it does come down to cost. There are many limitations with every study. No one study is ever going to cover every potential limitation, obviously. And that's one of the important things that we usually that we try to impart on our research partners and our sponsors, is that, look, what you really want to do is build a solid body of evidence, the totality of the evidence. Like if you ask me, would you rather have 4 small, well-conducted studies that all have small sample sizes and maybe you're underpowered in a few of the outcome measures, versus one mammoth, gigantic study where, you go all out and it's a $700,000 study? And I'd tell you, no, I'd rather have that reliability and reproducibility of being able to redo the study, even in multiple sites and centers as well, if possible. But yeah, so no, we do get a lot of opportunity to provide guidance and feedback on how many of these studies are run, at least in our CRO. That may not always be the case in others, but yeah.
Okay. All right. Yeah. What we hear a lot of the time about the cost of these studies kind of being a hindrance or a limiting factor in the scope or the duration or the population size. is there one or two things that you can point to as like, this is the main cost driver of a study, or is it more dependent on, I guess, the type of ingredients y'all may be doing, is the testing, or is it just, or without fail, every time you're running a study, this ends up being kind of like the most expensive facet of it?
Yeah, I mean, as you can imagine, it can vary dramatically. But most of the cost drivers, most of the time, are sample size. With all other things being equal, it's sample size, and then it's the number of actual direct contacts with the subjects, and the number of controls, and that usually means the number of visits to the lab. There's only so much you can do. We've done it all ways you can imagine, as you can. I mean, we've been involved, Tim and I individually and together have been involved in over 150 to 200 clinical trials. There are a lot of them that don't get published. A lot of them get kept under file for sponsors. We've even independently sponsored studies for ourselves that we just keep in-house as our own data. And that actually gives us a really big, I think, advantage. We have our own data set of mini pilot studies. We're actually up to over about 200 pilot studies that we've never shared that data with anyone. That's just in-house data that we keep. So anytime there's a new ingredient on the market, we secure sample, we get product in, we test, we run a small pilot battery, and then we end up keeping that data in-house. That gives us enormous insight that, to be honest, I don't think anyone else in the world really has access to. Even combinations like, have you ever wondered what a certain ashwagandha extract with a certain ginseng extract, standardized for ginsenosides and metabolites, what that does? We have access to data like that nobody else in the world does. And I think it's something that it gives us a unique perch and an opportunity to really provide insight. But getting back to your original question, it's really driven mostly by sample size and number of contacts, specifically even visits to the lab. And then that's something that contributes to cost with every study, regardless. And then you have other nuances like the actual study design. So is it an acute single administration, where most quote unquote nootropic energy products, for example, that can be a one single dose visit to the lab, and then you either cross them over or you do parallel design, right?
Yeah.
That's usually 3 or 4 visits to the lab. Or are you talking about a longer duration chronic training study, which really, really can be a bear, much more of a bear to run than people can appreciate unless you've routinely run studies like that. Right. These 8 to 12, 16-week studies where you're doing training, you're trying to control for training, diet, and then you've got the intervention. Your dependent variable is what you're looking for, and then you've got your intervention. So yeah, it's driven by a variety of things. But in our CRO, just to give you a range, I don't mind sharing a range. We've done studies and continue to do studies. We run about 25 to 30 a year. And we do, our studies can run anywhere between, say, 85,000 on the low end. Usually we don't do anything under that unless it's a pilot. Right. And then we do some work for some large CPG players that are in the 7-figure range as well. So that's a big range, right? So it's basically, 85 to 1.4 million. So it's a big range. Yeah. Wow. Yeah. But the majority of them do fall. If you run like a mean and a median, the majority of them probably fall in the 150 to 250 range for a higher quality study. You could run a study like that, probably a little lower cost at some universities, not all universities, right? Because some universities are very strict about indirect costs. And that can add an overhead of like 53 to 60% on top of the cost of the study because of indirect costs to the university, right? So the more prestigious the university, usually, as you can imagine, the more costly the indirects. Then there's some university that we also work with university partners as well, right? We collaborate there all the time. Some universities that have like an endowment or a foundation that allows you to run studies maybe a little lower cost. Sometimes they have undergraduates in the lab collecting data, for example. We don't have undergraduates collecting data at our lab, right? They're all professional, PhD, master's level, and MD or DO level staff at our CRO. So There's that, and then there's also lead times. That's big too, right? So there's a little bit of a running joke in that sometimes if in the time we've actually we've gotten IRB approval, enrollment, so screened, recruited, enrolled, and collected data for a full study. There have been some university partners that the sponsor has decided to also run the same study with, and they just barely got across getting IRB approval. Sometimes it's multiple months getting IRB approval. So lead time is important, right? And that's a business decision as well for some of these sponsors. I mean, do you want to wait 1.5 to 2 years for data, or do you want to have access to your data to be able to start substantiating your claims within 6 months, 3 to 6 months? So we're very fast. We're very efficient in that regard. That's why we're able to run 25 to 30 in a year.
Yeah, that's what it was going to be. I'm glad you brought up the university labs, because that was going to be kind of the next point that I was going to segue to, is that you're a private research organization. There's a couple of, better known or more well-known human performance labs, like the University of North Carolina, Chapel Hill's got a good one. Texas A&M's got a great one. University of Arkansas.
Yeah, Abby Smith is great. Rick Kreider out there, they're fantastic. Sean Arndt now, used to be at Rutgers. He's down at South Carolina, U of SC. Right. A few good ones in Florida as well. Yeah.
Is there I guess, aside from the possibility of undergrads or less trained individuals having a hand in the runnings of the study, are there any other major differences between using a CRO and a human performance lab at a university?
Yeah, I would say a couple of other things I would add to that. One is IP ownership can be pretty interesting. That can be a little difficult to navigate at the university setting, because as you probably know, most universities have a tech transfer office. Right. And so whenever you're running studies, it can be challenging to make sure, if I'm putting like my IP holder cap on for any of my ingredients, like SmartPrime, for example, or NAD3 or TeaCrine or Dynamine, if I'm running a study at a university, I do run the risk that if there's some new IP discovered, we may have to share ownership of that IP and licensing of that IP. Okay. It can be a little wonky the way the wording works in those research agreements. For us, though, we make it very black and white, very crisp and clear. We're here more as a business-friendly CRO. We understand the importance and the benefit of IP as an asset, and so we are unabashedly hand over any IP to the research sponsors that we work with. So any new IP that's discovered, any IP that may previously been unknown to the sponsor, even if we uncovered it, it all belongs to the sponsor. So that's one big thing that I think differentiates academia, from a privately held CROs. The other, we mentioned lead times, huge. I would say another, probably not so much with the principal investigators and the PhDs that run those labs you mentioned, because they're a little more attuned with how industry works. Right. They're colleagues of ours as well, and we're very friendly, and we attend meetings together often, and we're often collegial in sharing new data as well behind the scenes. But with many other academics that I've seen, they don't necessarily have the understanding of DSHEA or 21 CFR 111, and they also don't understand FTC, and they don't understand FDA structure function claim. So you can have a great study, let's say a joint health ingredient, right? And if you enroll patients, quote unquote patients, who have a ICD-10 diagnosis of osteoarthritis or rheumatoid arthritis, and those are your subjects, it doesn't matter how you couch the language on the results of that data. If you reference that study and you end up publishing that study, then the FDA interprets that as a misbranded, unapproved drug claim.
Whoa.
Yeah. So there are nuances, even on it's not just the patient population or their subjects, but even on the endpoints. So I've done some things where, I don't know if you've ever seen, like there's a big study we published on an ingredient called IUFlex. It's from Natrion, Terminalia it's a Terminalia extract.
Yeah.
And so that was a huge study. We ended up doing some really cool things there that were first in the industry. We looked at like spine wellness or spine health and comfort scores.
Mm-hmm.
But what I did there is we modified the WOMAC scale. So instead of using the Western Ontario McMaster Osteoarthritis score, mm-hmm. WOMAC, we did a modified. So it's literally called in the methods, you'll see it in the section. It's called an M-WOMAC. It's a modified WOMAC, modified for the natural products industry, essentially, because I scrubbed out all the drug disease language from there.
I guess what's the way you go about phrasing that? Because that's you brought on. You're naturally segueing to kind of everything that I've been wanting to hit so far. So this is a good little role we've been on.
Yeah.
So this is something that I'm curious too, because if you're sports nutrition is one thing, but when you start getting into the general health stuff, so cardiovascular support, joint health support, liver support, all that fun stuff, not as sexy as increasing your bench or your lean mass gains, but equally, it's not more important.
But you know what? I've convinced Tim over the years, and as he and I have gotten older, we now have also transitioned our CRO and our ingredient interest also into more general health crossover, right? Longevity as well. So
right. So when you're designing these things, so say you want to investigate a blend of like green tea and some other botanicals and see its beneficial impacts on cardiovascular function or reducing inflammation around the joints. How do you phrase that in the actual research? And then what claims how do you pass that on to the ingredient houses to say, hey, these are the claims you can make for the ingredient without getting in trouble with the FDA, or not getting in trouble, but getting a slap on the wrist or something?
Yeah, no. And that's exactly where you have to have one foot and we've worked with all the major legal firms in the industry as well. And I think that's a major benefit, right? I mean, one of my companies just does regulatory compliance separate from the CRO, right? Supplement Safety Solutions, where we do adverse event reporting. So we've been doing that for almost 10 years as well. And we've got such a good relationship as subject matter experts. Sometimes we provide expert witness testimony, or we're sort of a scientific opinion on behalf of many of the best legal firms in the industry that does this kind of claim language massaging. So we've become pretty good naturally by, with our work in the legal world as scientists, as scientific experts in the legal world. You have to sort of continue to wear a legal cap in terms of understanding where the risk is always exists along a spectrum, and understanding where there's a very bold line, or there's the speeding limit, and then there's how close to the speeding limit do you want to approach, or do you want to barely come over the speeding limit, knowing that if you do get pulled over, you're probably getting a ticket. So there's always an element of weighing and considering that aspect, along with what does the data tell us, what does the science say, and what do the results indicate, and how do we interpret that? So the results in the example you mentioned, in like a green tea extract for cardiovascular health markers or endothelial health, the results may actually say, let's just say you hit a couple of lipid markers that decrease, like total cholesterol, triglycerides. Let's say you were able to also improve blood pressure, right? Systolic, diastolic blood pressure improved. And then you're able to look at measures of endothelial function like FMD, like flow-mediated dilatation on brachial artery or ankle brachial index or something like that. It basically shows you've got improved, quote-unquote, blood flow. If you look at measures of like blood rheology, which basically measures a little more of like platelet activity and how red blood cells are moving through capillary beds. If you look at like oxygen extraction capability, things like we did with PurpleForce or the purple tea extract. So what happens there is you may be completely correct scientifically and have all the evidence you need to state in your manuscript things that would easily be drug disease language, that you're talking about cardiovascular risk factors, right? You're talking about cardiometabolic disease. You're talking about type 2 diabetes. You're talking about atherosclerotic risk factors, right? Or atherosclerotic cardiovascular disease risk factors. You're talking about hypercholesterolemia and hypolipidemic benefits. All those things, if you might be supported with the data you have in your research. And that saves you from being in trouble with the Federal Trade Commission, with the FTC.
Okay.
Because you might be truthful and not misleading.
Right.
But then you're in trouble potentially in the Office of Criminal Investigation with the FDA, because if you just take that language, that plain, clinical medical language, and you bring it into your marketing of that ingredient or that product, that green tea extract, now you have crossed the line according to the FDA. So you haven't violated anything FTC-wise.
Right.
Now you've violated FDA for sure, FDA structure function, Deshay language. You would be in violation of essentially introducing a misbranded, unapproved drug into interstate commerce just by making those claims that are truthful and not misleading. But you can't actually say the truth in your marketing, even if your science supports it, simply because the FDA has clearly delineated your lane, right? This is your drug lane, and this is your dietary supplement lane. And there are some clear demarcations between those lanes, but like when you're swimming in your lane, if you're doing laps sometimes, there are going to be some ripples and maybe some strokes that head over into the other lane a little bit. Like inflammation might be a great example, right?
Right.
You can mention inflammation only in the context of a non-diseased state or a physical condition, like related to exercise, related to day-to-day physical overexertion or something of that nature. Yeah.
So in those instances, you could say it supports so you could say supports cardiovascular health. Could you say reduces triglycerides, or is that stepping on toes?
No, you would have to qualify that. So you could say maintains maintains triglyceride levels already within normal range, already in the healthy range, or you can say maintains already healthy triglyceride levels. That's by most standards, that would be an allowable structure function claim. If you did a study that shows that you got statistical significance, strong, well-powered study that shows that your product did in fact decrease plasma, triglyceride levels.
Yeah. Okay. Yeah. I'm trying to think. I mean, it seems.
But then you can get creative in other ways. So we've done something where we've taken. Have you ever heard of something called like the predicts metabolic health marker, type 2 diabetes prediction marker? There's something else. There's a couple of these markers like metabolon releases and these clinical. They're used by physicians to basically try to predict risk. There's one that's run by LabCorp. When you run LabCorp and you do an NMR, like a more detailed, for people I may not know, that's more than just a lipid panel or lipid profile. You can run an NMR analysis on your fractions, on your lipoproteins, and really take a deep dive on like the particle size, particle number, LP little a. And then they actually have something that has 5-year risk data associated with your risk of developing type 2 diabetes based on this very deep dive lipid panel, which is called LPIR. It's a lipoprotein insulin resistance score. So you could take that data, and unfortunately, if you take that data because it's linked directly to a type 2 diabetes risk, you would be making a drug disease claim if all you did was just say reduces your insulin resistance risk score, even though your data supports it in your study. But what we've done is we've called it metabolic wellness, for example, improves your metabolic wellness index, or we've done things like that. You can get creative, and that's where sometimes you do need to sort of wear, your creative hat, legal hat, and, scientist hat as well, because you still need to interpret. Ultimately, that's the first step. And that's usually, I think, the biggest difference between someone who spends all their life in academia as a professor versus someone like what we do, which is we've been in academia, we've been in industry, we're running our own CRO, and we understand the needs, and we have our own IP and ingredients. So we sort of understand, and regulatory, right, with me.
Right.
So we understand all those different perches, I guess, that you might need to stand on, and those vantage points. Yeah, I think that's a big advantage.
Yeah, and on one hand, I understand why those regulations or those boundaries, the lane markers exist from just to protect consumers and, you know, from the nefarious players out there. Because there's whatever industry you're in, you're going to have, you know, disingenuous people.
Yeah.
At the same time, I also wonder, is that hamstringing ingenuity, innovation in the industry, or just the average individual being more willing to try some of these natural ingredients versus always jumping into the pharmaceutical route? And I don't want the listeners to mistake this. Like, I'm not against pharma or anything like that. I mean, it's like they certainly have their place. But at the same time, you know, berberine, for a perfect example, like berberine versus metformin, if we could say berberine, instead of just saying supports cardiometabolic health or supports already stable blood sugar levels, if we could say combats the risk of type 2 diabetes when we're marketing this one, it's got a considerable body of evidence behind it. You know, why? So I kind of wish
you're right
in that instance right there.
No, and you're 100% right. It does have that effect on the dynamics of how the consumer is going to perceive that product.
Right.
You know, it's going to almost like add fodder to the supplements aren't regulated, right? Who knows what's in that stuff, right? And it does, because you're hamstrung in what you can say. And the other thing is, when everyone starts saying the same thing, despite the fact that you've got maybe 3 or 4 or 5 different mechanisms of how they're doing it, right? I mean, look at the blood flow. You know, we do a lot of crossing over between the pump and non-stim pump and pre-workout space.
Yeah.
And obviously the maintaining already healthy blood pressure in the healthy range, right?
Yeah.
Hence, like VasoDrive being one of them. So you can play on both sides of that fence, but that's one mechanism. Then nitrates are obviously another mechanism, and precursors to the nitric oxide pathway, like the good arginines and the bioavailable arginines and citrulline, right, obviously plays another role, another pathway. And then you have the actual enos activators themselves, right? The polyphenols that actually allow you to prime the pump, if you will. So, it's hard to then be able to differentiate your ingredients sometimes when you're effectively hamstrung to only being able to say 2 or 3 claims, despite your mechanism and your data being really strong and impressive.
Right. Yeah. And so that's where, well, I mean, you get some of these questions every now and then, just on the different podcasts or in my inbox, you'll get something like, hey, what should I use? VasoDrive or Citrulline? I'm thinking, we're talking about 2 different things. Like,
I don't know.
Yeah, they have a similar end result, but putting them together is kind of where the secret sauce is there. And so,
yes, unless the consumer puts it on themselves, which it's a tall order, it's a tall ask, right? I mean, this is what we do on a daily basis, so we love geeking out about mechanisms, but it's hard for me to have a conversation with my cousin, corporate attorney cousin, or someone who just like, just tell me what to take, tell me if it works, is it safe, and I'll take it from there.
Yeah, exactly.
They don't necessarily worry about synergistic mechanisms and additive mechanisms. So
okay, let's pivot now to some of the actual ingredients. Do you have any specific one you'd want to jump into first? I mean, so SmartPrime is probably the newest one that has kind of hit the market. Do you want to start there, or do you want to start on one of the ones we've kind of maybe touched on a little bit already? Or
we could start there. Yeah, let's start with SmartPrime. That's fine. Yeah.
All right. Yeah, let's start with kind of a high-level overview. So omega-3 fatty acids, they're found in fatty fish. They're good for cardiovascular support, maintaining an already healthy inflammation response in the body, whatever we want to say about that. We're not going to get in trouble.
Right.
Most people don't get enough omega-3s in their diet. There's a great imbalance between omega-3s and omega-6s. So, people know that they need fish oil or omega-3 fatty acids. Where does SmartPrime-OM come in the greater cosmos of fish oils, omega-3 fatty acid supplements, and how is this, I guess, different or a better option versus what's currently available on the marketplace?
Yeah, so it's interesting. So the genesis of SmartPrime was actually a combination of, so one of my IP partners who kind of spearheaded the initial ideation on SmartPrime, Ryan Yates. He's actually a PharmD PhD, so he's a pharmacologist and a PhD in pharmacology. So a really brilliant guy, really gets into the weeds on mechanisms, on pharmacognosy and pharmacotherapies, and pharmacokinetics, et cetera, and pharmacodynamics. So where this comes in is we were looking at targeting metabolic disease or metabolic health. So both fatty liver, for those who may know it as different names. So NASH or NAFLD, non-alcoholic fatty liver disease, metabolic associated fatty liver disease, which, to be honest, I think is probably a more appropriate term because it's a little broader, more associated with metabolic dysfunction or dysregulation, insulin resistance, that sort of thing, combined with maybe some lipid issues, right, which somebody might already have genetically. So, it's interesting because if you look at a lot of the science behind omega-3 especially on like primary prevention, it was actually pretty disappointing when you look at some of the meta-analyses that have been conducted over the last 10 to 15 years. And I was a big omega-3 fan from just consuming more omega-3s, whatever it takes to get your omega-3 index up. Right. Because of a lot of my early work with Nordic Naturals, right? Nordic Naturals is one of the largest omega-3 manufacturers in the world. And
it's the bottle of omega-3s I got in the pantry behind me.
There you go. So and for many years it was the one that I also had and used with my daughters, even as they were growing up, and my wife when she was pregnant, and myself. So there was, on paper, when you look at all the mechanisms and all the pathways that omega-3s touch, it should almost quote unquote cure everything. But when you were looking at some of the data, it really wasn't quite, backing up what we would expect on paper. And there were a lot of disappointing headlines as a result. Now, granted, there were many limitations with some of these meta-analyses, even the studies that were included in those larger meta-analyses. As they often say, garbage in, garbage out, right? You have to be very careful about what studies you select to come into your meta-analysis.
Correct.
So often what was happening is, they weren't necessarily measuring omega-3 index effectively. Sometimes they were only measuring plasma omega-3, which is like a short-term snapshot of your omega-3 status, but doesn't really take a look at your red blood cell omega-3 or white blood cell plasma membrane, omega-3 index, which are important as long-term markers of your status. Sometimes they weren't using a high enough dose. Sometimes it was 500 milligrams. It was inconsistent, 1.5 grams, 2 grams, et cetera. But really the main limitation was that the genotype wasn't used as a way to randomize patients. And genotype has a lot to do with how you end up metabolizing, especially polyunsaturated fatty acids. It's one of those like major areas where you might have a SNP, and especially in the global population, there's really almost 2 alleles within the pathway of how you convert like the short chain omega-3 and omega-6 fatty acids to the end product, long chain ones, right? And so what we ended up doing was trying to figure out a way to sort of nutrigenetically modify that so that we would make all hypo responders, make them responders, and then make those who are already normal responders, make them even better responders, right? So going from poor to good and then going from good to great is what we were looking for with this IP.
Okay.
And so we screened multiple compounds. We looked at multiple bioactives. We kept coming back to the various sesamin isomers within sesame seed extracts. We screened multiple species of both white and black sesame seeds to look for the various compositions of the different lignans in there. And ultimately, we standardized the sesamin, but there's the secret sauce includes a complex of other lignans like episesamolin, episesamin, et cetera, in addition to sesamin. And then, so there was one is if you have the genotype that's consistent with someone from sub-Saharan Africa, right? It makes sense that if you didn't have access to the coast, you needed to be a very efficient metabolizer of those precursors in order to make that end product, right?
Correct. Yeah.
Because you weren't going to get preformed EPA or DHA, right, from fish anytime.
Right.
So you needed to be able to convert flax, perilla, ahi flour, all those precursors like that are high in ALA, for example, linolenic acid, to be able to convert that with the enzymatic machinery all the way to EPA and DHA.
Yeah.
And now oftentimes you hear a very, blanket statement like, oh, we're very bad at converting, those precursors to EPA, DHA. Right. And I was mistaken previously in that I also kind of just took the data for granted. And then as I dug in a little deeper, I realized, well, there's more nuance here. It really depends on your genotype. If you have a certain allele that makes you a hypermetabolizer based on how active your D5D and D6D enzymes are, and you're going to be very efficient even with a small amount of precursor. But if you're of Nordic descent, for example, you evolved having access to the coast, you had access to EPA, DHA, you didn't have to be a very good metabolizer because you were getting the end product already, right?
Right.
So you didn't have to take ALA or LA and make a lot of the end product, right? Because your body uses the same enzyme to work on both the omega-3 and the omega-6 pathway. And that's what many people, I think, don't realize. So if you're using the same enzyme to work on both omega-3 and omega-6 pathway, the end product, then it's going to be very dependent on what you're feeding it, right, early on, and what your enzymes are choosing to allow into the driver's seat to then turn into the end product, right?
Yeah.
And that's one of the things that SmartPrime does, is it allows you to the enzyme has two seats, one for omega-3, ALA, one for LA, omega-6. And it basically allows the seat to be much more amenable and comfortable to docking ALA, the omega-3, and it kind of crunches the other seat for omega-6 so that the LA does not easily come into the enzyme and allow you to flux through that pathway.
Interesting.
And that's one of the things sesamin and all of those related lignans do at a high enough dose. And, this can happen within liver, adipose tissue, skeletal muscle, but mostly in liver a lot in high activity, and adipose tissue. And then the other thing we were finding is that there's a phospholipid issue as well, which is if you throw a bunch of omega-3 at someone and just increase their omega-3 index, you don't know whether that omega-3 index, you don't know in what type of phospholipid that EPA or DHA is packaged in. So you don't know if that's packaged in a phosphatidylcholine form, like PC,
right?
Or is it in phosphatidylethynolamine form, PE, or is it PS or PI, et cetera, or the others? And really for just making the conversation more simple, you really want to have more in someone who's obese, overweight, has metabolic health issues, insulin resistance, cardiometabolic health issues. Usually the problem is they're packaged, mostly stuck and sequestered in the PE form, and not enough of it is going over into the PC form, the choline package. Right. So the first problem you have is, in most people, you want to get the pool up of omega-3. So you want more of that EPA, DHA, DPA, the omega-3s up there.
Correct.
And then now you've got the pool, now you have to package it correctly. That's the second issue. And that packaging does not happen in the gut. It actually happens mostly in the end cell, because everything gets disassembled during digestion. Right. So even if you consume like salmon roe, for example, which a lot of it is already in the phosphatidylcholine form, you're not just going to take that DHA molecule and the phosphatidylcholine form, or that LPC DHA molecule, and transfer it exactly intact across the gut into the lymphatic, into your blood system, eventually making its way to the liver and all your other tissues, or across the blood-brain barrier. That's something that happens. So once you consume the omega-3, it gets cleaved off, and then it gets reassembled and packaged in the small intestine, in the enterocyte, and then it ends up getting absorbed into the lymphatics, and then eventually, gets distributed to the body.
Fascinating. So is this something that would replace an individual's omega-3 supplement? Is this used in tandem with it? Is this kind of, is that where this is being more positioned on, like, hey, this is an add-on to make your body more efficiently, more appropriately harvest and utilize the omega-3s that you're already consuming?
That's correct. In theory, you could take this, and that's where some of the initial data was performed, both in animal models and in humans. And now we're going to be doing a much larger scale, a couple of human studies at larger scale, is that you can still take this in isolation, and it will, A, increase your omega-3 index, so increase the pool, and B, it will convert or transfer more of your DHA and EPA into the phosphatidylcholine form from the phosphatidylcholine. It will do it in isolation, but this is being positioned, and really it should be, as an amplifier, as an optimizer of whatever. It's agnostic to your choice of omega-3. So you can keep taking your favorite Nordic Naturals product, and if you add the SmartPrime to it, you'll have a better outcome. If you end up doing algae, better outcome. If you do flax, because you're strict vegan and don't even want algae, and you only do flax and perilla or ahi flour, you'll again improve and amplify the some of those endpoints regardless. And some of the other cool things that's happening is it's also increasing the efficiency of generating those resolvins and protectins, those downstream metabolites, the prostaglandins, leukotrienes, in the more healthy anti-inflammatory, anti-thrombotic, inflammation resolving pathways or signaling agents. Yep. So
delivery forms, is this in a powder? Is this going to be like at a gel cap or like a spray dried kind of thing? Almost, how are people going to?
It's a free flowing powder now, but it can be wet milled into soft gels, and it already has. Actually, there are a few products already on the market that are utilizing it in soft gel form. There's even one fairly substantial sports nutrition brand that's going to be making an announcement soon. So I can't really say who it is, but you'll definitely recognize them when they do. Leviathan already has it on the market in a soft gel form as well. But then there are a couple of companies that have also done it as just the powder in a capsule. And there are even some EPA DHA products, like Evonik has a VasoDrive-AP, which is like a lysine salt of EPA DHA in powder. And it's actually pretty high payload, because most of those powders are low payload.
Right.
It's actually like a 55 to 45% payload is a powder of EPA DHA. So you can do it in a capsule or a tablet with those as well.
Interesting. With these, the various ingredient and IPs, like, and I guess we can start with 3DPump, because that's kind of one of the newer ones on the block. From your mindset as one of the arbiters of the ingredient, do you like to approach it as, hey, we're going to blast this and offer to as many companies as possible, or do you want to be a little bit more selective? Because I know when like 3DPump was first coming to the market, I remember Bruce saying that, hey, we're going to offer only to a few select companies. Is that more ingredient dependent, or is like, for something like SmartPrime, where it's got to have a much wider range of appeal, do you want to go and blast it to as many people that are willing to try this new technology, this new ingredient out, or do you want to be a little bit more specific? And then do you, or I guess your business partners, restrict to make sure people don't pixie dust in like 50 milligrams of the stuff, or in the case of VasoDrive-AP, they're not going to put in 27.5 when you need at least like 254 or something along those lines.
Or above. So the answer is it's ingredient specific. You're right. Usually what we do there is you want to fit the ingredient to the first you want to find a partner, potentially a distribution partner, that where the ingredient ends up being a good fit for them, and then it's obviously a good relationship, a good fit for you in terms of their philosophy. Are they good stewards of QA, QC, GMP, and on the up and up in terms of supporting R&D and the science? Because that's at the very that's foundational for us, right? If you're not going to support continuous R&D, like not just resting on the laurels of doing one study, one and done, and then riding it all the way until the wheels come off, that's not our style. We're not going to end up being good long-term partners. But it's definitely become more, I think, initially with TeaCrine and Dynamine, I think that was, a very global blanket strategy from Compound to just try to, saturate the industry and get everyone to understand the merits and benefits of, basically pairing your products with TeaCrine and Dynamine. And now we've gone beyond, to be honest, even beyond energy and beyond caffeine and beyond neuroenergetics and beyond nootropics, because we've discovered some other non-obvious cool properties or features and benefits of TeaCrine Dynamine, including anti-inflammatory and more general health, even longevity, like affecting the sirtuin pathway and NLRP3 pathway and some other cool things that you'll be hearing some more about in the future. But yeah, I think it's ingredient specific. So with 3DPump, absolutely, my partners and I, Bruce and Tim, the three of us, we decided to be a little more judicious with it. And as a matter of fact, we're even able to pass along the savings of not having this go through a traditional ingredient house. So it's much more work for the company internally for us, but we feel it's worth it because we have a lot more control over the messaging, to your point, the dosing, and the brands that we feel comfortable having the ingredient associated with as well.
Going forward, and I mean, I guess this may be painting too broad of a stroke, but do you like having the partnerships with the ingredient house or distributor, or do you kind of like, now that you've got a taste of doing it with 3DPump, do you kind of like being the master of your own destiny kind of thing, and you and your partners control where the ingredient, the supply, and all of that stuff?
Yeah, I don't think there's a preference right now, to be honest. Both avenues bring with them, various trade-offs. There's some pros and cons to both. When you're doing it on your own, you obviously have potentially a little more risk, a little less support. You also don't have as much of the outreach necessarily. So that creates a little more burden on some of the infrastructure. When you partner with an ingredient house that already has that reach, that brings some other benefits as well, right? You're spreading out the operational risks, you're spreading out the logistics, you're also spreading out the financial risk. And then the way we do our deals in that regard, it's much more of a JV, much more of a joint venture, because we even though we stay behind the scenes, we are pretty intimately involved in the direction, especially in certain things where like roles and responsibilities, like we definitely handle in terms of any R&D sort of responsibilities or roles. It mostly ends up, ultimately coming through us, through the IP holders, whereas sales and marketing, we leave that up to people that know much more about sales and marketing than we do, right? So
with the ingredients, we don't see a lot of head-to-head comparisons of this. And like, I guess in this avenue, since I got you here and you've kind of spearheaded a couple of things,
creates like confusion. No, you're right. Yeah.
Well, with like, say you want to run a study and you want to say, hey, TeaCrine versus Dynamite versus an extra or something like that, and you want to see energy increase, or you want to compare 3DPump versus just regular citrulline versus nitrosogene or Sulflo 6 or something like that. Do you stand at any kind of, or you or the company, I don't know how you want to phrase it, at any kind of legal risk by just comparing somebody else's ingredient to yours, since you have a vested interest in it? Or say your ingredient comes out on top and you just say, hey, look, we were 2 times better than this ingredient.
Than a specific branded.
Can you do that, or can you not?
Yeah,
I'm curious why we don't see some of these kind of studies more often.
I think you can do that, but you definitely increase your legal risk substantially by doing that. And I think that's probably the major reason why you don't see more of it, to be honest. So we're more, I think, I think a happy medium is looking at, like, for example, we're doing with 3DPump currently. So we're already enrolling and collecting data on patients, right? So it's 3DPump at 6 grams versus just straight L-citrulline at 8 grams. So you're really comparing 3 grams of citrulline inside 3D,
right,
with 8 full grams of pure citrulline. And it's a generic ingredient. That's a great ingredient in and of itself, right? There's a reason why we use it in 3DPump, right? It carries many merits with it. And I think most of your listeners are very well aware of, why citrulline is included and so pervasive in the sports nutrition world, right? And even beyond. But we just used it a little differently. And I think there's some of the nuances probably gets lost when you look at the panel of 3DPump and you're like, oh yeah, it's like an admixture of 3 ingredients that we've already known for some time. There's a little more nuance there, obviously, and it's part of the IP, right? We screened over, 12 different botanical ingredients, and until we came up with the one that ended up really showing us synergy. There were others. To be honest, it surprised us. We thought there'd be no difference, for example, between a maritime French pine extract, a pomegranate extract, a grapeseed extract, as long as it was rich in those paranthocyanidins and some of those polyphenols. We thought that they'd probably be very little difference, but that's what IP is about. When you get a pleasant surprise and then you reproduce it, and sure enough, it's there and it's there again, and it allows you to make, you've observed something that's non-obvious, and it's more than just phenomenological, and you have something that's novel, non-obvious, and synergistic, and you've got real IP. So that happened with us with 3DPump, but then we also don't use silica as a carrier as much anymore as we used to with the original OG glycerol HydroMax that eventually paved the way for everyone else. Once the trade secret got out after we licensed it to Glanbia, which I must credit, actually, a lot of that started with, I believe you had, we texted briefly about this. I think you had Dan Pierce on your show, right? On a podcast?
Yep. Yeah, about a month ago now. Yeah.
So Dan Pierce introduced us. So I don't know if you mentioned it, but he introduced us to Glanbia way back when. Yeah, he knew about HydroMax or what we were doing. It was called something else at the time. When he was with Gaspari in his first year or so, he was there. And he made that introduction. And we took it from there. We ran it up the ladder to the science folks at Glanbia. They really liked it. And so we ended up, licensing it to them, and selling them the IP. And then eventually it got out, some of the everything we went through 15 different silica types before we found the one that worked. The inlet temperatures, the slurry, the outlet temperatures of the nozzle, of the atomizer. There are a lot of little details that go into why even HydroMax, with all its problems, if you've ever seen HydroMax as a standalone, we have HydroMax samples from, I kid you not, in our offices. We have HydroMax samples from 2010, 2011 that's still free-flowing powder.
Really?
In isolation. Yeah. The issue is, obviously, when you have augers that introduce heat and friction, when you have other hygroscopic ingredients, the nitrates especially, any free bases like L-arginine, hydrochloride, anything that drastically changes the pH, even high acid loads of citric and malic acid. Things like even betaine, as innocuous as it might be in isolation.
That's super hygroscopic.
When you start mixing all these ingredients that each have their own hygroscopicity to them, it's only a matter of time before they cause a problem. No matter how much someone claims that their glycerol product is stable, it's glycerol. Unless you've changed it, chemically, then it's no longer glycerol. Then it may not clump, right? It may no longer be hygroscopic, But then it's probably not going to do what you want it to do intravascularly anyway, right? So.
True. Going back to the polyphenol source y'all identified as being the most optimal for inclusion in 3DPump. So it's the Indian gooseberry, the amla fruit extract. Is it so? We know that these polyphenols, they're all at the core, they're stimulating endothelial nitric oxide synthase, which is the enzyme that's going to, catalyze the conversion of arginine to nitric oxide for the listeners that may not be sure when we're throwing around these terms, what all's going on. So is it a matter of assessing the potency? Because like you mentioned, grapeseed's got some fun polyphenols in it. Pomegranate's got the punicalagins in it. And, you could have even used maybe like a citrus sinensis peel or some kind of orange peel extract, because we've seen ingredients come out that have those fun bioactives. And so we've got all these different types of bioactives. Pine bark's another one. Is it a matter of just like which one is doing it the greatest amount for the least amount of bioactive compound that you need? Is that what you're kind of I trying to identify with this? Because they all have the same result, but maybe you only need 3 milligrams of this where you might need 300 milligrams of this other polyphenol source. Is that kind of where like the secret sauce is in trying to identify which one?
Yeah, so you have to almost like pick your limiting variables. You have to kind of do it systematically. So for us, we usually do it based on total milligram of a product. So for grapeseed extract, we used a 95% OPC, standardized product, right? For some of these others, you're not going to get a 95% active. So you're not going to get as, so you're talking about maybe measuring some of these polyphenols that are much more playing a single note, they're only playing one instrument versus the orchestra of all the other family of flavonoids and polyphenols that might be included in a less isolated, less standardized mixture, right? So one of the things we did is we looked at like a 20% low molecular weight tannins, a 40%, a 65%, and an 80%. And for us, we actually found that a middle ground of 40% ended up bringing many, many more results that we didn't see with a more concentrated version. And the reason we felt that happened is because low molecular weight tannins in this case are doing more than just working on enos. There's probably other compounds that are non-tannins, that, like not the alagotannins, for example, and not the low molecular weight, monotannins, but they're probably other flavonoids that are maybe even there's some catechins in there, for example, that at a lower level are acting more directly on the enos enzyme, on the endothelial nitric oxide synthase enzyme. But what's happening with the tannins is they're actually working more on the actual RBC, on the red blood cell rheology. So they're attaching to glycoproteins. That's been found mechanistically in an animal and in vitro model, actually, with Indian gooseberry extracts and low molecular weight tannins and alagotannins. They actually interact with the RBC, with the red blood cell itself, not with the enzyme lining your blood vessel. So it's a completely different mechanism. So in theory, you could combine a low molecular weight tannin, which we didn't really appreciate. You could combine low molecular weight tannin with, for example, like a pycnogenol or a pine bark extract. And you might potentially see something more additive. It may not necessarily be synergistic, but it might add something. It might bring something to the table, to the party, that you otherwise wouldn't have. But what makes the low molecular weight tannins unique is they do a few things acutely, and then they do some things long-term that gets better over time the more you take something like the 3DPump, which is why we're going to do a second study on a long-term adaptation study. It's doing some things to recovery in advance, for example, of your next session. So it's almost like if you take it intra-workout or pre-workout, it's almost already helping to amplify the recovery and adaptation process before your next training session.
So almost something like a tart cherry extract or something, like there might be some kind of polyphenols that are supporting maybe some
modeling or DOMs or something.
Something like that allow you to recover faster, and then you go and train. You can train more frequently and more intensely.
So there's something else happening with the glycoprotein. So the rheologies where we feel the magic is here is the glycerol is adding plasma volume intravascular. Then the citrulline is clearly providing substrate for ultimately allowing you to manufacture more and make more nitric oxide. For the enzyme, the nitric oxide synthase, more arginine, and then eventually more NO. So you got NO, you got plasma volume, and then the low molecular weight tannins of the amla, the Indian gooseberry extract, the philanthus, is doing something to change the rheology of the red blood cell that allow it to flow through capillary beds more efficiently. And it also ends up doing something over time where those can convert in the microbiome. They can be biotransformed into urolithins, urolithin A, B, C, and D, some of these urolithins, which we know there's actually some ingredients now showing potentially mitochondrial health improvements from the urolithins as well over time. There's even something with the low molecular weight tannin that makes it act also as a mild ACE inhibitor as well, like VasoDrive, also very mild. So it's really doing a lot of heavy lifting in many different areas.
Mm-hmm.
In any one of those areas, it may not be a heavyweight champ, but it's probably more so, I think, what it's doing with the red blood rheology acutely that allowed us to see these acute benefits in terms of how much blood and plasma was able to shift intramuscular from the intravascular to intramuscular compartment.
Mm-hmm.
We did these acute DEXA studies, and when we did these BIA, multi-frequency segmental BIA, to look for fluid shifting into muscle tissue from blood. Yeah. So.
Wow. Cool. Yeah, I was some like I've talked. I talked to Bruce before and Aaron, and it's just that's one topic that like I've talked with Aaron over at Performax about this because he uses 3DPump, and just like he sent me some of the initial patent research on it and stuff like that you guys had sent him, and this is the first I'm hearing about the red blood cell rheumology. That's pretty cool. I'm gonna have to go and dig it. Like that's something I've not even heard before.
As an engineer, I'm sure you'd appreciate fluid dynamics and rheology. So
that's fluid dynamics. That's you're gonna give me nightmares about college again with that class. Good Lord.
Yeah. So I think there's more going on than meets the eye. But look, I think it's hard to expect every brand owner that brings the ingredient in to understand like all of those nuances, right? Yeah. It's probably going to get lost on the consumer, which means, as a brand owner, it's probably not as important to them as well anyway, right? So.
Correct. Yeah. This is more for the people that just love diving down, ingredient mechanism wormholes and things like that. So.
And honestly, Robert, I think part of what happens is, again, it's like the nuances and the context is important. We have access to being able to do these things in a controlled setting. It's not just like the bro, like, hey, let me take it to the gym and give it to like 5 guys and see how they feel. It's like, it's not a controlled setting. There's so many variables that can confound your results, right?
Right.
And unfortunately, sometimes that's how brand owners make a decision. I'm not faulting it because that's great for us to be like, that's where we start, like ourselves, hypothesis generating, right? Anecdotal data. I mean, it's where you start generating hypothesis, but ultimately you still need to explore that and ask those questions in a more rigorous fashion before you can really make a determination as to whether something's like complete, it's like, oh, this is bullshit. Like there's nothing novel here. It's I apologize for
you don't have to worry about there's anything goes on this. This is not a kid-friendly podcast in any way, shape, or form.
Good. So I mean, you could see it and say, well, what's it going to do beyond citrulline at 3 grams? Well, we'll show you, because clearly we wouldn't be running a full-scale study that's going to cost even us over 6 figures ourselves out of pocket in order to be able to compare 6 grams of 3D to 8 grams that only has 3 grams of citrulline compared to citrulline in and of itself. There's also just the pragmatic aspect of it too, with supply chain being where it is. We have all these ingredients locked up. We've had them locked up for a long time because of other stuff we do in the industry, right? With HydroMax, obviously, et cetera. And so we were able to come up with, I think, a pretty good all-in-one solution. If you're just looking for, like, let's say you put a pre-workout together and it's like, all right, I've got one pump ingredient, quote unquote ingredient or ingredient matrix, one pump ingredient, and then one stim. What are you going to do? Well, you could do caffeine and 3D pump, and you've got a pre-workout potential.
Yeah, you're hitting at a couple of different points along the way and everything. Plus, you could get into, some of the beneficial pump aspects that caffeine in and of itself it has with vasodilation, calcium release, and all that other fun stuff.
Yeah, planking stuff. Yeah.
Let's get into some more general health. Say it again.
Yeah, you want to cover some other ingredients or some other stuff?
Yeah, let's do one more with NAD3, and then I think I will have kept you for a pretty long time at that point, and we can table the rest of those for the part 2 of the podcast.
Sure.
Let's do NAD3. This is a topic we have not really covered a whole bunch on the podcast. I talked to Sean Wells about a year ago. I think it's coming up on with some of the stuff that he was doing with NMN and NNB nutrition, but we haven't done a ton on just NAD, nicotinamide riboside, NAD just in general. So, let's start with a little kind of a high level overview for the listeners about why is NAD important, and if they're listening to any other kind of supplement related podcast or just like longevity, fitness, wellness stuff, they've probably heard about the importance of NAD, NAD plus, NADPH, all of those kind of things. So I guess let's start there and delve into what NAD3 is and why it might be a little bit different than some of the other NAD related substances on the market.
Yeah, so I think high level overview is there's been some seminal work in the NAD space in general that basically put it on the map and got it reinvigorated, re-excited beyond it just being a byproduct of vitamin B3 metabolism of niacin, right? We know that NAD, for decades, we've known from a biochemistry standpoint that NAD is an essential coenzyme that allows you to move around electrons and hydrogen atoms, basically, in the majority of redox reactions in the body and most living organisms, to be honest with you. It utilizes NAD or NADH. So NAD plus is the quote unquote oxidized form of nicotinamide adenine dinucleotide, the nucleotide NAD plus. And the NAD plus, the reason it's plus, it's charged because it's missing its hydrogen, and therefore it's been oxidized. It's missing an electron, and that's why it's charged positively as NAD plus. So that's where people see NAD plus. NADH means it's been reduced from the whole redox differentiation, reduction oxidation. It's been reduced, and actually the electron and the proton have been paired now, and now you have NADH. And so it can go back and forth and sort of play ping pong, or whatever other analogy you want to use, like pass the balloon. If the hydrogen and the electrons, the balloon, you're passing it along back and forth to be able to transform molecules and reduce them or oxidize them throughout the entire body in every single cell of the body. So we already knew NAD was essential for life. We know that without NAD plus, without a sufficient NAD status, you probably couldn't live for more than a few seconds at a time. It's that important. But then I think the paradigm shift came when we realized that NAD plus itself could also be used as a fuel or a substrate, like a raw material that's used as fuel for other enzymes, where it actually gets used and broken down and degraded. The way you would almost break down and degrade glucose, for example, right? And convert it into something else, completely degrade it. So now you're not using it for redox reactions to move around electrons and hydrogens, right, in the mitochondria or in the cell. Now you're using NAD as a substrate, as a fuel, because it has some components that the enzyme is going to then use in order to signal to the cell to change its metabolism, to change how it's turning on or off genes, to change which genes are being repaired, how DNA is being repaired potentially. So if you want a cell that's going to be resilient ultimately to a lot of environmental stressors or a lot of things that we see in normal Western, or everyday cultures, whether it's due to environmental pollution or pollutants or overconsumption of calories, alcohol consumption, overweight, type 2 diabetes, et cetera, you're going to tax the NAD system in the body. And NAD plus is a substrate for specifically the two main enzymes that we know it's a major substrate for. One is called the sirtuin family, which many people have been following the longevity space have probably heard about already. And there are 7 different sirtuin isoenzymes in humans that we know of. And then there's something called PARPs, or poly-ADP ribosylation polymerases. Basically, these are things that take the NAD molecule and borrow something from it in order to tag it to other proteins in the cell. And the sirtuin uses NAD as what's known as for its the enzyme sirtuin. What it does is mostly deacetylate things. It removes acetyl groups from proteins, from histones, from other compounds in order to change how genes are either being expressed, like to turn on like resilience programs or programs that are considered, they make a cell more resilient to the effects of aging, accelerated aging. So over the last 8 to 10 years, there's been a big movement in finding ways to augment your NAD plus status. And that has opened up this whole, sort of, I guess, wave of various methods to try to improve or increase someone's NAD status. Yeah. And that's anything from, Chromodex's product Niagen or NR, nicotinamide riboside, to the NMN molecule that, again, you mentioned NMB has, and David Sinclair has talked about. Charles Brenner is the scientist that held a lot of the original IP on NR that ended up being acquired by Chromadex. And so one of the things that there's a bit of a war between NR and NMN, you're probably familiar with. To be honest with you, one of the things that intrigued us is I felt like there were a few things happening. I felt people were missing the higher, higher order, higher level point here. Like from the top down 30,000 foot view, you should almost be agnostic to how you get your NAD levels up. There are many ways to improve your NAD status. Yeah. Exercising, maintaining calorie restriction at the appropriate times may help you also increase your NAD status, but exercise is a great way to do it, especially intramuscular NAD levels, which, to be honest, as of right now, we don't have good evidence that any supplement increases intramuscular levels of NAD yet. Most of what we have seen in the data shows that if you supplement with NR and MN, sure, you can reliably see increases in blood NAD levels. Right. That's from PBMCs, or basically blood cells within the blood. Or if you just look at whole blood levels of NAD, if you just take the whole blood and analyze it, you can see NAD plus levels increase. Okay. So what we did is my partners and I took a look at what was happening and said, there's a few things missing. One is, what if we weren't just worried about just pumping more precursor into the system? Because if that's the point, why couldn't you just use niacin or NMN or niacinamide, to be honest? Right. And actually, there's data showing that you can increase blood, liver, and actually in people that have mitochondrial myopathy in human clinical trials, niacinamide and niacin have both been shown to increase muscular levels and help normalize muscular levels of NAD plus in muscle with 250 to 500 milligrams of daily supplementation, even over as little as 4 to 12 weeks. So you just got to deal with the flush that comes on with the. Yeah. So what I do personally is I prefer to overweight it towards niacinamide. So I use a little bit of niacin and niacinamide in a blend, basically. That's my NAD precursor of choice. But I don't mind NMN. It works. Kind of pricey to do it compared to niacin or niacinamide, but it can work. Right. NR can work as well. So basically, we took a step back and said, okay, what if we were agnostic to whether you use NR, NMN, niacin, or niacinamide? Is there a way for us to look at the actual pathway, like the biosynthetic pathway, the NAD metabolome? Like how does the body make NAD on its own? Yeah. Well, it makes it through a few different pathways. It can take tryptophan and do de novo synthesis, very complex. There's probably about 8 to 12 different chemical reactions, biochemical reactions to get to NAD. Then you have something called the Price-Handler pathway, which you can take nicotinic acid or niacin. That's another word. Niacin or nicotinic acid are interchangeable. Right. You can take niacin and make NAMN, and then eventually get to NAD through basically 2 or 3 enzymatic steps. Okay. Then there's the big one, which is the way most of our cells in our body make the majority of our NAD, is the salvage pathway, which is recycling either dietary precursors like taking niacinamide or niacin, or what happens when you take NAD and break it down by having it be consumed by those enzymes like sirtuin and like PAR, the PARPs. When they consume NAD, they break off niacinamide and it can be picked up and recycled again. So that's the salvage pathway. So look, the body's, it's incredibly intelligent, and it's so redundant, so many adaptive pathways. There's so much redundancy. we didn't just, discover a way to allow our bodies to get enough NAD, right? In the last years. So the issue there is just providing the body with enough precursor. And there are many pathways. There are many roads that lead to Rome, many roads. And you can look at this if you'd like, go to a Google Images map and you look at NAD metabolome and you'll see all the pathways and you see they all get to NAD. So we said, I wonder if there's a way for us to upregulate the enzymes that are controlling those steps, right? To be able to take any of those precursors and be a little more efficient at making more NAD if you need it. So if we were able to accomplish that, then we knew that we basically could be agnostic to whatever your choice was, and you could use our ingredient with any of those precursors. But beyond that, the other thing I looked at is I wanted to look at all of these health span promoting and lifespan promoting behaviors or activities. Things like, even if there's some controversy over whether intermittent fasting works because of just calorie restriction or whether there are some unique benefits. It seems like if you parse out the data, there might be some unique benefits with truncating your window. And even early, if anything, early time restricted feeding would probably be the more optimal choice. Things like the Mediterranean, a well-constructed Mediterranean diet, exercise, whether it's high intensity interval training, whether it's long steady state duration, zone 2 to zone 4 training, whether it's strength training, all of these things, including, polyphenols and all these, hormetic stressors, heat stress, sauna, mindful meditation, all these things have a signature. They all have a molecular and biochemical signature. So if you perform any of these activities and if you were ever able to assay what's happening within certain tissues and certain cells, you could see that they all have a unique biochemical or molecular signature, right? So there's like an orchestra of these events taking place. So what if we could maybe amplify that signature or mimic that signature? So that's the other thing we were looking for in trying to screen various compounds to lead us to NAD3. The third thing that was the linchpin for us that allowed us to discover NAD3 was we started seeing some unique things with TeaCrine. If you go back to the 2016 paper by Lem Taylors and the group out of University Mary Hardin-Baylor, the 2016 paper, it was the safety study where we looked at 200 milligrams for a month of TeaCrine versus placebo versus 300 milligrams. We even went above the grass dose just to show safety, even beyond grass, even beyond our 100x, grass dose, beyond the NOAL. And we still showed that there was something that was non-obvious there, that signal that was statistically significant, but it wasn't a primary outcome. And we're like, that's interesting. We saw a statistically significant decrease in total cholesterol, LDL cholesterol, and a trend in triglycerides in both the 200, and it was like dose dependent. So it was stronger in the 300 milligram group, but we were already seeing it in the 200 milligram dose group over just 4 weeks. Interesting. And these were like healthy college age individuals that were starting with like a total cholesterol of 185 or 180, 190 maybe. Yeah. So that was something was going on there. I was like, I wonder what's happening. Could that be, hepatic, liver, mitochondrial, something going on? So we started sponsoring some animal work. Some of it was done in China. And we started doing some in vitro work that was published since. And you'll actually see a trail over the last, 2016 is probably 6 or 7 years. Yeah. If you look up TeaCrine and like longevity or TeaCrine and sirtuin, TeaCrine and mitochondria or LCAD, like long chain acyl dehydrogenase, you'll start seeing where we stumbled into something non-brain related, non-energy related to TeaCrine. So TeaCrine is one of the ingredients in NAD3. It's 3 ingredients. It's NAD3. Right. It's a wasabi that's very rich. We screened multiple wasabi sources. It's very rich in a unique blend of isothiocyanates. These cyanates are found in like cruciferous vegetables. So everyone's probably heard of like the broccoli, like glucoraphanin and sulforaphane, right, is the end product from these glucosinates. These are all families of isothiocyanates. Moringa has it. A lot of these other cruciferous brassica vegetables have it. Wasabi, I've always been a fan of wasabi. Love it. I can't have a sushi meal without extra wasabi. Right?
Oh yeah, same here. Same here.
You like it? Yeah. So, so I always felt like intrigued by it. I was like, I wonder if we take a deeper dive and see if there's some other bioactives that we can look at. And so there's some like methyl phenyl isothiocyanates. There's some unique families of these compounds in there. And we started doing some in vitro work of like, if we pulled out, for example, wasabi and put in another botanical like broccoli, broccoli extract, if we pulled in quercetin, for example, if we pulled in some others that we knew might have some longevity mitochondrial benefits, would we get the same synergies we were seeing with combining TeaCrine with it? And we weren't. So there was a unique signal when combining TeaCrine with wasabi that we ran into in vitro, with all other conditions being controlled. Granted, it's in vitro. Obviously, there's a lot to be able to go from in vitro to human, but it was that in combination with what we were seeing with TeaCrine. And then I've also always been intrigued by the concept of copper. Most of the way we supplement our copper is using cupric, or the +2 oxidized copper form, even the copper chelates. But the type of copper called cuprous nicotinate, which ironically enough uses niacin as a way to chelate and maintain copper in the +1 valence state, which basically means it's just it's got a difference in the orbital arrangement in terms of it's a +1 instead of a +2 charge on it. And it's the way that the body uses copper in a lot of these like sodium oxide dismutase enzymes. And it's how it uses copper as a cofactor, mostly in mitochondria, as well as in some of these other antioxidant enzymes. So we combine these three ingredients: a mineral, a botanical, and an alkaloid, TeaCrine. And you put it in various ratios. This has been about, a 5.5-year process in the making, running it stealth, not really telling anyone until about 2 years ago when, we still semi-stealth, ended up partnering with a couple groups to launch it. And now we're in conversations with some pretty substantial brands to license it. So that's an example of where we took an ingredient, took a very different licensing approach and commercialization approach. We just finished publishing our first human study. We have another subset human study that's coming as well. So we actually supplemented in a very well-controlled. We were pretty rigorous if you look through the methods section, how we controlled for it, placebo versus NAD3. And we actually measured NAD levels, even though we weren't supplementing with a niacin precursor at all. We still were able to see improvements in the NAD status, probably because of those enzymes that we saw upregulated when we did like transcriptomics. We did RNA-seq to look at like all the genes that were being transcribed and turned on or turned off in cells. We saw some of those genes being turned on and amplified. And we also hit like, there's this within the longevity geroscience world, like of aging, there's these 9 molecular hallmarks of aging. Lopez Ortin, unrelated to me, a different Lopez out of Spain. They published some work which talked about the hallmarks of aging in 2013. We actually have shown that mechanistically in preclinical models and or in human clinical models now, we're able to hit almost all 9 of those molecular hallmarks of aging. Everything from a pathway related to cellular senescence, stem cell exhaustion, intercellular communication, epigenetic regulation, telomere integrity, mitochondrial health. So we're excited to hit all 9 of those hallmarks of aging. So we took a different approach than everyone was chasing after, like senolytics like Fisetin or NMN and NR. We just said, you know what, you can use all of those, but we're going to bring you something that, much like SmartPrime, is meant to be a companion ingredient that can elevate like the rising tide that raises all boats in that space. So you can use it in conjunction with any of those other ingredients, and probably additive or interactive benefits. And we're doing a study now, which is pretty cool design, where we took elderly subjects again, and that's the other thing, is this is the target population. So we're not looking at this in 20-year-olds, 50 and over, 50 to 65 plus. And we're doing a study now where we're actually using a unilateral resistance training for legs method. Mm-hmm. And we're taking muscle biopsies of both the trained leg and the untrained leg at baseline. Ooh. Yeah, I know.
So, like, you got 50-year-olds to sign up to get a chunk of their muscle just ripped out?
And we're using an academic partner for that one. Yeah, we've done a fair number of biopsy studies at our lab, like as a CRO. Man, it's definitely it's not easy. It's not easy, and it's not easy to recruit and enroll patients, right?
Right.
But they get good compensation for it. So it's not quite inducement, but it's a healthy compensation and stipend for their contribution to science.
There you go.
Yeah. So we're getting biopsies of both legs pre, and then post training, and then we're also getting blood. So we're going to try to see if what we saw in the first study in humans, where we only looked at blood and safety markers, and we saw like gene expression changes, we saw telomere changes, NAD, we even saw those lipid changes, again, confirmed, but even amplified with a smaller dose, because we're using less than 200 or 300 milligrams of TeaCrine in that blend. And now we're going to look to see, can we see changes in muscle itself? And then we're going to see do we have performance changes, functional capacity, changes in some of the other important metrics of health span and lifespan beyond just like, oh, did you move some esoteric gene signal like sirtuin in blood or in a muscle cell, right? So
fascinating. Since you're studying this in an older population. Is this, and just in general, I guess this applies to most of these longevity supplements, is it really worthwhile for 20 and 30-year-olds, even maybe early 40s, to even think about this kind of stuff? Or is it something like, is this a supplement that really you're not even going to see the utility or justify the expense until you're maybe past 45 or 50?
That's a great question. And based on everything that I've seen and my experience as both a clinician when I was practicing for 10 plus years, in spine and sports medicine, right? Work with athletes at all levels, from amateur to pro, and work with elderly individuals who have sarcopenia, right, and beyond. I think that what's interesting is most of what we're trying to do from a supplement intervention for improving or supporting longevity, I think probably has very little merit or utility in the very young population, because the young population already they have such a low inflammatory, like cytokine like milieu in their plasma. They have such a low senolytic burden still. Their mitochondria is still so healthy. Everything is working like, the way you want it to, right? So I see very little benefit, to be honest, with anything that we're doing currently in terms of intervention. But having said that, there's two caveats to that. One is lifestyle change has a much more important impact when you're younger, to get you to a peak level of, just like we used to say in women, right? You should try to attain a peak level of bone mineral density or bone mass before you're going to get the inevitable decline,
right?
Into that osteopenic, osteoporotic stage if you live long enough, right? In an estrogen depleted state. I think the same thing we should have the same approach for skeletal muscle mass accrual. I think you should try to accumulate within reason as much of that peak level of skeletal muscle or lean body mass as you can before you start seeing the decline. So before you're 35, Right. And then I think beyond 30 to 35, depending on what your biological age is, which when we get a better biomarker of biological age, there's some promise now with some epigenetic markers out there. Companies like TrueAge have a really good one that we actually used in our human study, looking at epigenetic age and epigenetic profile. And biological age markers like phenoage, like Morgan Levine's phenoage, which we're measuring in both of these human studies coming up. I think there's probably like much more merit to using it once you're starting to see some of those measures just at the point where you're starting to see some of those other biological age markers start coming up a little bit. Yeah. So probably 30 to 35 would be the youngest. I think that there might be merit for that. Yeah.
Yeah, and that's where I was trying to get. I get at, and you touched on it. Is that sure? It might have like an infinitesimally minuscule amount of benefit when you're in your 20s, just because you said like everything's still running in peak shape. Assuming you're doing the things like you're eating well, you're sleeping well, you're not stressed, and you're training hard and all that kind of stuff. You're not drinking yourself to oblivion every otother day of the week or something like that. And so just because your body is naturally doing all of these things, is it more like you're getting the spillover to where you're supplementing with it? It might have some tangential benefit, but nothing you're really gonna realize. Not something as maybe necessary as like joint health support or something along those lines, which you will probably want to start that early and then continue as you get older to attenuate any further declines in structure, function, inflammation, those kind of things. So
I agree. I think what you do is you move the big blocks, the big boulders first when you're younger. Yeah. One thing I would start really early is get your omega-3 index optimized. Get your omega status optimized. Make sure you're following good, like we said, good dietary principles, stress management, sleep, exercise, etc. And different exercise modalities, not just cardiovascular, cardiorespiratory, aerobic HIIT, but combining it with strength training and even starting to address mobility and biomechanics as well. Yeah. But I think it's just get your big boulders in place, like vitamin D status, magnesium status, omega-3 status, sufficient protein status. Get your 1.6 to 2 grams per kilo per day from the time you're in your teens, if you can. If you can get those big boulders in there first, even creatine monohydrate. I mean, I started supplementing, I guess I was probably 17, maybe, EAS. I guess Creatine was the first time, the first creatine monohydrate supplement I started taking in the early 90s, maybe. I've been supplementing for, well over 35 years, 30 years. I've been on 3 to 5 grams of creatine, even before the all loading versus maintenance, stuff came along.
Yeah.
So, yeah.
And your kidneys haven't fallen out yet? You don't have chronic kidney disease from creatine supplementation?
Believe it or not, right? Yeah. And obviously, as you probably know, I'm a co-author on some of those position papers on safety and the data, right, for creatine. So, yeah. I actually used creatine when I was at Northwestern at residency and got it on the formulary at a lot of the Chicagoland hospitals. Cool. I did a study in chronic stroke patients, actually. Mm-hmm. We did a randomized controlled clinical trial. We had stroke patients that were 6 months out from their stroke. So we didn't have that like weird area of spontaneous recovery where everyone's getting better after their stroke. Right. So 6 months stable post-stroke, but they still had their hemiplegic gait where they had one side of their body was clearly affected and motor deficits. And it was affecting their gait and their ability to walk, more than, a couple meters per second, for example. Yeah. And we did a creatine group, small scale. It was 7 subjects in each, arm. Creatine versus placebo. This was 2004, 2005, 2005. And we did body weight supported treadmill training where they had a harness and they were walking over the treadmill to try to improve their gait status. That was the only exercise they did for 25 minutes, 3 days a week, both groups. The creatine group saw dramatic improvements we were just doing maintenance 5 grams a day we only did 45 days of training and supplementation. Their improvements in not only gait speed, but like we didn't even train squat, but their ability to do chair sit to stand, like 35-40% improvement over placebo. Their ability to negotiate stairs, to go up to climb two flights of stairs. And then what we saw then that I should have pursued further, I just got busy and went into private practice and didn't pursue it further. I didn't even publish it. I just published it as an abstract in the grand rounds, sort of internally within Northwestern and Chicago. And this is where it was interesting. We saw improvements subjectively, like VAS measures in mood, their perceived ability to focus, their perceived ability to perform cognitively and cognitive function. Yeah. So they were they felt happier. They felt they were in a better mood. And already we were getting early indications that creatine is probably one of the oldest nootropics, right, that we've had.
Cool. Well, yeah, because there's been like the last 4 or 5 years or so, there's been a a boon of research about the cognitive aspects of creatine.
Yep.
I mean, it's awesome. It's like my wife's been on it for a couple of years now. I, like every now and then I'll sprinkle a little bit in my 4-year-old's like smoothie or shake that I'll give her or something just to we'll start micro loading it, and we'll, when she gets a little older, I'll hit her with the main doses of it.
I did that when my oldest, my 17-year-old, when she was she got her black belt in TKD and Kukiwan Taekwondo, World Taekwondo Federation. So when she was starting to get to a point where some head shots, for prophylaxis from head injuries. So
yep.
That was like 7 or 8 years ago, and I had her on like she was at 3 grams a day probably for her size.
Yeah, and probably for that size, that's more than enough to maintain saturation or achieve it and then maintain it at that.
Yeah, man. So
awesome, Dr. Lopez. We are almost at the 2-hour mark, and you and I were talking for a good 10 minutes before we even officially started this thing.
Yeah.
I don't want to abuse your time any more than I already have.
No worries.
Thank you so much. This has been a masterclass in supplement science, research, structure function claims, like legal stuff to keep people safe with all that, the ingredient deep dives. And there's still so much more that I want to ask. I'm going to table out
ingredients to review and more, right?
So much more. Plus,
and even ingredients that I'm not directly involved in that I could give you, an opinion on based on our database, like I told you, of over 300 studies, 250 studies that we've conducted. Yeah,
exactly. Is there anything you want to plug or mention, or things where you want people to reach out to you or anything like that?
No, I mean, I don't do much social media. I mean, I really don't. I used to do more Twitter. I used to be involved early. I don't know. Maybe now that Elon's
taking over, you're going to jump back on.
Maybe I'll jump back on again. It's tough. I'm involved in a lot, both within the supplement space and even more now I've ventured more into the biomedical health space and doing a little more in health span and longevity and health and human performance from midlife on. For individuals. Just joining more boards, doing more stuff in board stuff. I'm also an advisor to multiple private equity groups as well as a domain expert. So LinkedIn probably is the easiest way to get in touch with me, I'd say, if somebody wants to reach out. And I look forward to chatting some more. I know it's tough to get the schedules to work, but we'll do it again. This was fun. So
absolutely. Awesome, Dr. Lopez. Thank you so much. And we'll catch you next time.
Awesome, man. Thank you.